Hi, hello, and welcome to this session on biological agents, which meets the learning requirements of ND2 1010. So I'll go straight into it. So by the end of the session, you'll be able to outline where biological agents are likely to be encountered in the workplace, how these can be controlled, and also a section on infection control links to COVID as an example of a biological agent reaction. So I just wanted to talk first of all about the special link with COSH and the L5 requirement. So I'm just going to screen you that one at first, but what I want to do is show you the screen for that. Yeah, so let's go to the previous one, Regulation 2 I want to look at in particular. Yeah, so using the ACOP L5, you've got mentioned in the regulations like application and the various areas that COSH comes into practice. And then you've got links to hazards that are related to all kinds of COSH aspects, but a section on biological agents in that definition. So the point I'm trying to make is it's worth getting familiar with these aspects, not just of the regulations, because you can see it's very, very detailed. It's all still the regulation at this point. And then when we get to mention there, where a biological agent has an approved classification, any reference in these regulations to that particular group in relation to the agent shall be taken as a reference to the group to which that agent has been assigned and in that approved classification. And then you go further into the guidance then supporting of the aspects of various parts of it. I'm not going to go down to it, but if I go back to the beginning, I want to click on in the index Schedule 3 just to show you link that, so that when you've got a copy of L5, the code of practice supports COSH regulations, you can see the specifics there around additional provisions related to work with biological agents. So interpretation of schedule initially, the part that plays in regard to biological agents, then classifications. Okay, so when you go down to 2 there, and then you go A through to D, you can see the 4 groups based on level of risk. So group 1, unlikely to cause human disease. Group 2, can cause human disease and may be a hazard to employees, but unlikely to spread to the community. Group 3, can cause severe human disease and severe impact. And then group 4 causes severe human disease and is a serious hazard to employees. It's likely to spread to the community, and there is usually no effective prophylaxis or treatment available. So you can see how the severity increases there. The idea behind this is to consider the level of risk associated with it. If you're dealing with any of the biological agents that fall into particularly groups 2, 3, and 4, then the hierarchy of controls and the levels of controls tighten up. So as you can perhaps see in the next section when it says special control measures for laboratories, animal rooms, and industrial processes where biological agents are present. So you can see there, as it's the schedule, it has a special legal standing. It's almost like reading directly from the regulation itself, therefore treat it as the law. every employer who is engaged in any of the activities specified in the subparagraph 3, which is just above there, biological agents, shall ensure that measures taken to control those biological agents and exposure are adequate or suitable combination of contaminant measures from the list. So then it gives you, or the person that's handling, as the responsible person, the approach that they need to consider. And in some respects, you could argue the licensing requirements to comply with handling of groups 2, 3, and 4. So the nature of the activity, the containment levels, the risk assessment, and the nature of the biological agent concerned. So all those aspects are vetted in order to gain approval from the health and safety executive to handle these biological agents in a safe manner. So think about what happened with COVID, the potential debate, should we say, that's going on around whether it was in fact produced in a laboratory, and unfortunately because of the reduced standards, should we say, control measures, perhaps that's how it escaped and therefore started to have an effect at group level 3, where society had been seriously impacted, and therefore vaccinations, isolations, etc. are used as a reactive measure. So I'll be talking a little bit more about that later. So I just wanted to share that one with you as a starter, because I think it's worth putting into context when we consider the COSH regulations in regard to biological agents. So you can see there, mentioned it in relation to his session with the regulation and the description that goes with it. And believe me, it's quite detailed, but we'll go into the hazards and then the expected control measures in the next few slides. So, types of biological agents. Microorganisms are bacteria, viruses, fungi, or even parasites. These microorganisms are covered in the COSH regulations and classed as biological agents. They're defined as any microorganism, cell culture, prion, or human endoparasite, whether or not genetically modified, which may cause infection, allergy, toxicity, or otherwise create a hazard to human health. So focus rather than on the detail there, but focus on the final statement there of saying hazard to human health. Due to exposure to the biological agent, it results in impacts on humans. So typically we would look at organisms and entities that present occupational health risks: fungi, bacteria, viruses, protozoa, algae, and endoparasites. So thoughts around that will expand, but we tend to focus on control measures around these 4 areas. So the principal types: fungi, bacteria, viruses, and protozoa. So we'll look at those in more detail now. Fungi. Any member of the group of eukaryotic organisms that includes microorganisms such as yeasts and molds. They can cause infection on or inside the human body, for example, athlete's foot, on, as a classic example there. Some product toxins that are harmful to humans need to be considered as well, that could have an impact on the reproductive activities of human beings by forming spores that are released, dispersed, and found in a suitable environment to grow. So warm and damp, typically. So farmer's lung is a typical example of this, and it's caused by breathing in the mold, and then that grows on the hay, straw, and grain. So it's around farmers, and therefore when they breathe that in, it's like occupational asthma, and in more severe cases can be fatal, similar to cancer-causing effects. Bacteria as a biological agent. Humans and most other animals carry millions of these simple single-celled organisms. It's around the body. No nucleus is present in the cell. Some can move through liquid, some form spores that can survive heat, cold, and lack of water. So it's not ideal for them, but they can still grow in the right environments. Bacteria inhabit soil, water, acidic hot springs, radioactive wastes, and the deep biosphere of Earth's crusts. And a classic example of this is, if you think about Legionella as a bacteria, it's in so many water areas all around us, but usually in small amounts. And it has to be in the exact conditions for the bacteria to grow to a harmful amount. So think about systems in the workplace whereby Legionella can, or the bacteria, should we say, can grow. It's stagnant water, should we say, typical examples of that, where it's got warm temperature, just above the surface of the water, that is allowed to be idle and therefore in some respects stale. And if it's not moving around and there's nothing like ultraviolet light to kill it off or other means of filtering it out, then it can grow to substantial levels. Viruses, self-replicating molecules that invade host cells, take control of the cell to produce more viruses and then release these viruses to repeat the cycle. Hence the escalation, should we say, through the body. The problem with this is because it's so small and its affinity to the body, it relies on susceptibility, should we say, and the body's immune system. There can be a period of illness before the body's immune system becomes effective. So the classic example in that regard is the common cold. Eventually, our system builds up immunity to it and fights it off. Severe viral infections, classic example of smallpox, can cause death. And therefore, we hope that it doesn't affect us nowadays, but it's still around the world, there are still examples of it taking place. And then viral infections not dealt with effectively by the immune system because the immune system itself has broken down. So a classic example of that is HIV. Protozoa, a single-celled organism with a cell nucleus, able to move and do not typically photosynthesize. So it's limited, should we say, in its strength and ability to spread. Diseases caused by protozoans include malaria, sleeping sickness, giardia, amoebic, dysentery, and toxoplasmosis, just to name a few. Treatment is by specific drugs. Antibiotics are only effective against bacteria. So it's limited in that respect around antibiotics. So you've got to specially treat it, should we say, to support the immune system, because unfortunately the immune system is unable to deal with the diseases caused by protozoa. So let's look at some examples of pathogenic causes linked to biological agents and where they come from, and then how it can infect and affect human beings. So it could be a pathogen that's transferring from one human to another. It could be interaction, again, with humans and animals, or even animal to animal or bird to bird. Then you've got pathogens around plants. Allergic reactions are classic examples of that in interaction with humans. And then finally, the environment, thinking about water sources, aspects of land, movement through contamination around Earth in itself because of the environmental ability to transfer from one area to another. So let's look at each one of those. Humans, to start off with, fungal, bacterial, and viral infections are passed from person to person. They transfer typically through bodily fluids. Viral hepatitis is transferred by needle stick injuries. Droplets infection, this can be, as an example, tuberculosis, spread by the droplet made when people cough, sneeze, or even talk. Again, similar to the idea of CV19. Physical cross-contamination, such as flu viruses that spread by others touching a contaminated surface. Again, very similar to COVID-19. Let's look at the interaction with animals and the potential for biological agent transfer there. So fungus or fungi, bacteria and viral infections passed from the animals to the humans in the same way as person-to-person infections. That are a bit more specific, should we say, to animals, and therefore we classify that as such. So the viral disease rabies can be passed by a dog biting a person. The bacterial disease leptospirosis by coming into contact with water or surfaces contaminated with an infected animal's urine, rat urine typically, and then zoonosis. So a zoonotic disease, those that can be passed from non-humans, usually a vertebrate, to the human. So bats and the likes of. Plants. So some plant diseases are caused by viral and bacterial organisms. Most are caused by fungal or fungal-like organisms. Sign of plant disease, physical evidence of a pathogen, fungal fruiting bodies, which is always an interesting one to look at in terms of its symptoms. It's worth doing a bit of research on that just to see the impact and changes in the body as a result of that exposure. Symptoms of plant disease, visible effect of disease to the plant. Yeah, so it'll change its state on the plant, look a little bit like mold. It'll change in color, shape, or function of the plant as it reacts to the pathogen. Similarly, from that point, it's transferred to people and therefore has an impact. So a classic is pollen. So direct contact, pollen, or insects can pass the pathogen to a person. That exposure to Pseudomonas aeruginosa, typical to, or similar to, Legionellosis, and the types of, so waterborne and movement around water areas, but it's ingested. So a classic example there, rather than just the obvious pollen examples. Found in most environments and can lead to inflammation and sepsis. The environment. So severe occupational diseases can originate in the environment and have the ability to transfer. So here we've got Legionella bacteria in the right environment, right temperatures, damp soils, water courses, etc. The Legionella bacteria can grow to significant levels. So that was just to give you a flavour of some of the typical groupings and typical concerns around biological agents. Now let's move on to the link to COSH in regard to being able to familiarise with different chemicals and properties, but also then to think about the ill health effects and then further the expected control measures to manage and reduce the potential risks associated with those exposures. So, differ from hazardous chemicals as they have the following special properties. Yeah, so its ability to reproduce, rapid mutation, the incubation period. Hence, that's why when testing takes place, quite often it can get 3, 4, 5 days, or sometimes 2-3 weeks before it starts to fester, should we say, and build in the right conditions. It's obviously got that infectious property, its ability to transfer into the body and through many people, and then the rapidity of the multiplication of the virus or bacteria, but the biological agent itself. So you can see here why it's specific to biological agents in comparison to other hazards covered by COSH. And therefore, because of this affinity to people and the impact it can have on health, the necessary controls must be to a higher standard. So let's look at each one. Rapid mutation. The genetic code changes over time. They mutate the characteristics of the organism, and that changes. Some biological agents have a very high mutation rate. The genetic code and characteristics can change quickly. They can get difficult for human immune systems to effectively recognize and attack. And that's one of the key concerns, should we say, the body's immune system, its ability to attack if, like HIV, the attack system or the immune system is itself not compromised. So think about that in terms of AIDS, the links to HIV, and how the mutation rate is significant, very high mutation rate in that particular example. Incubation period, so delays between infection and the first signs and symptoms. Again, from a disease that's growing, it's very awkward to try and pinpoint where it exactly came from, because mutations can take quite a while and variations on that. So incubation period is about time as such, from days to years, and that's ability to do that. Creutzfeldt-Jakob disease is an example of this, where it could be a long time, 30 to 50 years, before it comes into full fruition. So it's about time, and I said, as a significant time, the presence of a biological agent may not be readily detectable, and finding a link between the original source of the infection and the symptoms of the disease may not be possible, because it's been such a long time since it first mutated. Infectious. Most diseases spread by making the carrier infectious. Think about the host, who is going to be carrying that infectious amount, and then how it can be transferred. So who then poses a risk to anyone they come into contact with. Example of this, as a biological agent, tuberculosis is a highly infectious disease that can be spread within a community. Group 3 and 4 in some respects. Legionnaire disease is not as infectious, could be class group 2, as some would say, but quite often group 3. It's not as infectious as a disease, and it's not passed from person to person. Is asymptomatic aspects around this, a carrier for a disease or infection, but experiences no symptoms themselves. Again, thinking back to COVID, there were a few people, particularly the younger end of society, didn't seem to show the symptoms associated with COVID-19. So similar here, hepatitis C, some people can have the infection and totally unaware of it, but they are a spreader of the disease. Rapid multiplication. So this ability to be able to start the movement, and you've probably seen it under microscopic growths, where it starts off as 1, then it multiplies to 2, to 4, to 8, to 16, and so on. So the rapidity of that multiplication is an interesting factor to consider in terms of a property. Suitable environmental conditions allow microorganisms to multiply very rapidly. For example, E. coli. And it lives in the intestines of healthy people and animals. So we may have it in our systems for a while. Some people could be asymptomatic, as was mentioned before. But it also is linked to contaminated water and food, and can multiply at a rate of approximately 1 cell division every 20 to 30 minutes. So if you think about how food can degenerate due to bacteria, it comes down to this cell division and its ability to just rapidly multiply. A few bacteria that might not be present at risk of infection can multiply quickly to a large number that can overwhelm an immune system. So can the body respond effectively and quickly to this multiple growth? Even with limited exposure to biological agents, the risk may be high. It's not the simple dose, but its ability to multiply rapidly. So the simple dose response equals the response outcome from the exposure to the chemical substances, biological agents, and also the immune system. Let's look at a common one. So zoonosis, or the zoonotic disease, it has this ability to pass from non-humans, so vertebrate typically, and then move on to humans. So there's quite a few examples of that, but tend to always think about bats and their ability to transfer disease of this type. Approximately 300,000 people in a variety of occupations are potentially exposed to approximately 40 potential zoonoses in the UK itself. So that's quite a concern, should we say, but In proportion to other biological agents, it's quite small. Typical examples, Coxiella, chlamydia, and toxoplasma. Coxiella, Q fever. It's a bacterial infection caught from infected farm animals, sheep, cattle, goats, by close contact with placenta, blood, urine, feces, and animal skins, fur, and wool. So very small bacteria, and it has this link to an ability to grow around those blood, urine, feces, as well as sitting on the surface of the animals. Quite often the carriers are asymptomatic, but others typically show as flu-like symptoms within 2 to 3 weeks of exposure. So chronic Q fever can lead to serious heart problems. Endocarditis is an example of that. Chlamydia, reports of abortion in farmers' wives following infection with ovine strains of Chlamydia psittacia during pregnancy led to the study of the distribution of chlamydial antibodies in rural populations in northwest England, where endemic chlamydial infection with abortion is common in sheep. The parasite can remain for toxoplasma in soil in a cyst form for long periods of time, spread by the ingestion of water, food, or soil contaminated with feces from infected cats, consumption of undercooked meat, pork and lamb typically, and contact with infected sheep during lambing. So toxoplasma is quite a worry for farmers and handlers of animals and stock. Exposure may occur in those who are in contact with infected cats or cat feces, are in contact with infected sheep during lambing, or work with materials or products from infected animals. Lots of different vector-borne diseases associated with zoonosis. So anthrax all the way through to streptococcus. Just give you a chance to read through each one of those. So zoonotic vector-borne diseases. Human illness caused by parasites, viruses and bacteria transmitted by vector infection. So the picture on the left there is showing a bite, and as a result of the bite, as much as the insect is trying not to deliberately cause a disease, it's feeding from the blood, but unfortunately it's a pathogen. So as it's biting and taking the blood in, its infection is transferred through that movement. So the vectors are living organisms that transmit infectious pathogens between humans or animals to humans by the bite of infected arthropod species. Mosquitoes is a typical example, but we've got ticks, triatomine bugs, sandflies, and black flies. Lyme disease. A tick-borne bacterial infection that can be transmitted to humans when they are bitten by an infected tick, found mainly in the south of England and the Scottish Islands. Only a small minority of ticks in the UK are infected with the bacteria that cause Lyme disease. Being bitten by an infected tick does not necessarily result in Lyme disease. Again, it comes back to the affinity, should we say, and susceptibility of people that are bitten. Commonly, this presents as a spreading bullseye rash around the bite area, and it is quite evident. It stands out, you've got that central darker bit, and then around the outside, a lighter section, and then another ring around that. About a third of cases in the UK do not notice a rash and may present with fever, headache, fatigue, or neurological symptoms. Quite a nasty one, that though, Lyme disease, and awkward to treat as well. Occupations then that we typically consider to be at risk. Obviously, people who handle all kinds, should we say, of animals, and in some cases links to exposure to insects as well. So farm workers, vets, zoo workers, pet shop workers, sewage workers, and construction workers. Again, because they're disturbing the land, they're open, they're outside, and they may be coming into contact with certain insects as well. So just to make sure that there's an awareness there about sewage workers and construction workers, which don't come obviously to some people. Just going to now show you this video on infectious diseases.
In this next chapter, I want to take up the subject of zoonoses. A zoonosis is an infection or an infectious disease that is transmittable under normal and natural conditions from vertebrate animals to humans. It may be enzootic or endemic or epizootic or epidemic. There's also another term, zooanthroponosis, which is a disease transmitted from humans to animals. Now, there are different types of transmissions with zoonoses. There is direct transmission, that is, intimate contact with an infected animal, a person, such as with a bite, a scratch, a spray by infectious urine, contact with fecal material, inhalation of a discharge, respiratory droplets due to coughing or sneezing, airborne spread. This is direct transmission. Then there's indirect transmission, where there is an arthropod vector, such as a flea, a mite, a mosquito, a tick, and fomites, such as cockroaches, that are contaminated with the particular product going from an animal to a human. Another term we should be familiar with is species jumping. This is the transmission from animals to humans, followed by human-to-human spread. And this is the group that we are most interested in. That is, we shall see with HIV/AIDS, with SARS, with flu, that the particular agent went from an animal to a human and then began to spread from human to human. So the first one I'd like to take up is SARS, or Severe Acute Respiratory Syndrome. If you'll recall, this completely paralyzed the world a number of years ago because the mortality was quite high from this. Well, it turns out that, that SARS, as we found out much later on, was due to a particular virus called a coronavirus, which was found in a particular animal called a civet cat, which was slaughtered in some of the markets of China. But these civet cats had, in fact, been infected by bats. So it was bats that had the coronavirus that infected the civet cat. The civet cat, when they were killed, the spray of blood, sometimes people inhaled, they got it, and they were then able to transmit it to other humans, and it led to a type of pneumonia. And if you'll recall, there were often pictures such as this from China, which is where the disease particularly had a major impact, although it then spread to Hong Kong and Singapore and Canada and to Vietnam and so on. It never became the global pandemic that some of these conditions can become. This is the latest of pandemics. This is the Ebola virus. Many of you have heard about this. It's been particularly devastating in West Africa in this recent outbreak, but it has occurred in Central Africa as well. Let's take a look at this particular virus, which has been considered very contagious, though not very infectious. That is, it can spread from one person to another through contaminated feces and blood and so on, but doesn't spread from person to person in a crowded room, for example. It doesn't spread through the aerosol route, but rather through the blood and feces and so on of individuals who are infected and sick. They have to be sick with this disease. Here are some of these workers wearing these gowns to prevent them from having contact with this particular virus. This virus has probably, though we're not sure, been spread by fruit bats. That is, the bats, again, carry this particular virus. The bats may have contaminated food that was then eaten by an individual, and that individual developed this disease, Ebola, and then spread it to other people through their bodily secretions. But it was the bat that originally harbored this virus, then spread it, gave it to people, and it was through the person-to-person spread that it has taken off. Let's take a look at another one of these zoonoses. This is one called Nipah virus. Quite interestingly, again, this giant fruit bat, and why they harbor these viruses, we're not sure, and it's very uncommon actually, would eat some of the fruit of these trees in Malaysia that were planted in areas where pigs were grown. The pigs ate the fruit that fell to the ground, and because this fruit was contaminated by the virus, the pig, in eating the fruit, developed an infection. When these pigs then were taken for slaughter and killed by in the abattoirs, the slaughterhouses, the men who were involved in this, in the pig killing, got the infection from the pig. So it went from a bat to the fruit to the pig to the people who were killing the pigs, and up to 250 people died from this condition, a mortality of around 40 or 50%. And this occurred in 1998 and 1999. And to get rid of this particular condition, first of all, when the pigs became sick, first thing people did is they took them to market. So oftentimes these sick pigs, they had developed a pneumonia, were taken to market, and people would buy it unsuspecting that this might be this particular condition. And the way that Malaysia eventually got rid of this Nipah virus is they took pigs and they killed them. And there is no longer a pig industry in Malaysia, where at one time it was quite flourishing. This now takes us up to Bangladesh. And in Bangladesh, there was also an outbreak of Nipah virus. And they couldn't figure out how did this virus get into the human population, because there were no pigs in that part of Bangladesh. It was a predominantly Muslim society and pork was not eaten. But they pieced together a story that showed that when these bats went to this date palm juice, now here's a pot on a palm tree that is collecting date palm juice, which is a sort of sweet juice that can also be made into something called gur, which is a type of sugar. And when they would lick this, because they're fruit bats and it was very sweet, and sometimes they would defecate into the pots. The men who sold this, and this date palm juice was considered quite a delicacy, would take it from house to house, and sometimes this date palm juice was contaminated, and individuals who took it would get Nipah infection. And the mortality, again, was around 40 or 50, sometimes 60%. This was a meningoencephalitis, that is, it affected primarily the brain. Here are some bats flying just at dusk, and they're quite large. They have a large wingspan, but they only drink fruit and sweet things. So the way to get rid of this was to put a skirt of bamboo over where the sap came from the tree into the pot. And by doing this, you can actually reduce the contamination of the date palm juice, because the bat cannot have access to that sap that's coming from the tree. So again, a bat to the fruit, contaminating that, humans ingest it, and it can spread from one person to another only on very, very intimate contact. And this is only through respiratory droplets. So it's not like someone can walk into a room and cough and others would get it. You have to be extremely close to the individual to get this particular infection. So how do we control the transmission or the introduction of zoonoses? Well, this diagram here shows the linkage between wild EID as emerging infectious diseases, domestic animals, and humans. And you can see that they interact. Now, I'm not going to go through this entire graph, but let's just take a look at a few of the linkages. The linkage, say, between wildlife and domestic animals. Here, if domestic animals are closely associated with certain wildlife, as we saw with the Nipah virus in Malaysia, there's a spillover effect, and these domestic animals can pick up the infection. If we look then down to the bottom of domestic animals and look at the food processing technology industry, we see that this virus got into humans through the infection in pigs, which was then picked up in the slaughterhouses. And then in humans, it spread from person to person. So we have to break a number of these links between wildlife and domestic animals, between domestic animals and humans, and even between humans and wildlife. And so a lot of the strategies that have been developed to control zoonoses have focused on this type of diagram that we see here. All or most of the new and re-emerging diseases, not all, will in fact be related to zoonoses. So zoonoses are clearly very, very important and extremely costly. Bovine spongiform encephalitis, or mad cow disease, costs the UK over $9 billion in lost revenue. The Nipah virus, which I just talked of, cost Malaysia $540 million because of all of the slaughter of the pigs that had to take place. SARS, which we started with, cost China upwards of $50 billion because of the lost revenue from trade and tourism and so on. And avian flu, H5N1, which we have not covered, is going to cost billions and billions of dollars. In fact, another type of flu, H1N1, or what was called, unfortunately, swine flu. This is just a picture of the economic impact it had in Mexico, where this beach would be filled with tourists at a particular time of year. And of course, it's empty now because people are afraid and go elsewhere. What I've tried to do in this brief time is to talk about the importance of zoonoses, that is, the diseases which linked animals to man, and how they can spread, how they develop. SARS, Nipah virus are but two examples, and there are many, many others, and then how they might be controlled and the economic impact that they can cause. We must be prepared to put much more attention into zoonoses, and there is a global program called One Health, which is trying to do just that.
Makes you wonder what the true cost of COVID will be when you look at the economic impact associated with that globally. Anyway, let's move on. As a result of the level of risk from biological agents, there has to be some control in place, and therefore cost regulations set this out in some respects, but the higher the risk requires more tighter controls to be in place, and therefore licensing requirements for handling of biological agents. So additional control measures may be required for general and incidental exposure to these biological agents. Giving in examples, animals, infectious people, handling waste, material contaminated with microorganisms, etc., and working in sewers. So obvious controls tend to fall in line with a hierarchy. If in this respect, if you can't avoid the exposure to it, eliminate it in some respects, then you've got to think about how then, if it is in the air or around the areas of contact, how can it be minimized or controlled or reduced. So avoiding exposure is always the top approach and priority within a hierarchy of this type. As an example, can use cameras rather than going down a sewer. Training, information instruction, so that people are fully aware of not just the hazards but the control measures expected and the rules that need to be obeyed because of the level of high risk. Suitable disinfection procedures, so that as you transfer from a contaminated area, maybe there's a dirty area where things are taken off or washed down, and then there's a clean area whereby you can then change into controlled environment, should we say, where you can change back into your own clothing, and therefore minimize the potential for spread. Arrangements for safe collection, safe storage, safe transportation, and safe disposal of contaminated wastes. Vaccinations, as can be found from COVID-19, and the requirements there perhaps to reduce the potential impact as a result of supporting the immune system. So known biological agents cause zoonosis type conditions. Personal protective equipment and respiratory protective equipment, again, as has been said in other aspects of COSH controls, you can't really do it without this. So as much as it's considered to be low down on as a risk control measure, typically it's kind of an expectation as such to prevent breathing it in and also coming into contact with viruses, bacteria, and forms of biological agents. Hygiene, as you probably saw from the video there, about wash down procedures, and as I've just been saying about moving from a contaminated area to a dirty area to a clean area. Yeah, so having those procedures in place to prevent or reduce the accidental transfer of the biological agent, typically including provisions of appropriate and adequate washing and toilet facilities, and also prohibition of heating, drinking, smoking, and applications of cosmetics in those areas where the biological agents are present. Just going to try and show you the link to this, but employees who work abroad, this will work, and show you what I'm trying to do. Really want to bring this up. Bob and Bodrum, yeah, like that link. Bob and Bodrum. Teresa shying away from Turkey. Lovely picturesque area, but staying away now because of the impacts or potential transfer of disease in those areas. Okay, so I want to take it a little bit further now and thinking about infection and then linking this chain of infection and things that can go wrong, and then considering how we can perhaps reduce or minimize the impact of it. So the chain of infection looks at the process of infection can be represented as a chain. It breaks a link in the chain at any point, then that will stop or reduce the potential infection. So if you look at source, transmission, and host, if at some point you can do something with the source, it doesn't then transmit to a host. Similarly, if the transmission or method of movement from the source to a host is stopped, that will break the chain. And then finally, if the host is kept away from the source and limited chance of getting contact with transmission, then it stops the chain of infection. So when you identify the hazard, you need to find out about the links to the chains to help you identify the best way to break it and ultimately control the risk. So there are four main approaches that we use for this in the workplace. Firstly, blood and other body fluids and sources of blood and body fluids, such as human bodies, animal carcasses, and raw meat. Also, if you're handling or in proximity to these, there's a chance of that taking place. From personal experience, I look at the time I spent at court funeral care, and embalmers are particularly at risk from the deceased, whereby handling of the body and the movement of the, should we say, the liquids from the body, potential contact with skin themselves. And again, it goes back to the idea I was mentioned before about, suited and booted, almost like a hazmat approach, gloves on as PPE and RPE in some respects, and then moving from the area where the biological agent is to then go into a dirty area for wash down before you take off your personal protective equipment and Wellington boots, etc. So all that's washed down in the dirty area, and then you can go into the cleaner area where the removal of the PPE takes place in between the dirty and the clean. And then again, it could be sterile or cleaned down as appropriate. And then finally in the clean area where you can change into your own clothing. So there are aspects of that, procedures that need to be in place to break the chain. Secondly, human or animal waste products such as feces, urine, and vomit. Yeah, there's quite a lot of biological agents associated with movement around feces, urine, and vomit, therefore problematic, particularly for children or vulnerable older people as well. Respiratory discharges, coughs and sneezes, and then finally skin, known as direct contact. So we've got to think about the microorganisms such and look at its affinity in air and its living capabilities, and then the interaction that it can then have and that movement through the chain. So infection at work can occur via person to person, indirect contact, hands into the mouth and nose and eyes, breathing in the infectious aerosols or droplets, plus splashes of bloods and other fluids, body fluids, broken skin where it's injected, shall we say, and got through the broken skin, and then finally a skin penetrating injury. So those are typical approaches to how it can get into the body, and then we have to consider what does the body's response to that in terms of the propensity for infection to take place. So infection can be transmitted person to person or animal to person, and these diseases, as you found out earlier, zoonosis. Now, third point on the right-hand side that you can see, the conditions need to be right for infection to transfer. Yeah, so it's as awful as it's been sounding, all the earlier information there, it really does have to fall into the right category: the temperature, the humidity, the affinity to the tissue, etc., etc. So all of those can impact on this microorganism's ability to move around. So if you can interfere with it or stop the chain by hygiene processes and other methods, it can make a significant impact on controlling spread. So let's take that a bit further. When we consider breaking the chain, hosts or reservoirs are a place where germs can live and multiply. So we have to think about where those areas are and perhaps minimize and treat them, should we say, as higher risk areas. The place can be a person, a patient or a client or a member of staff, but it can also be any part of the surrounding area, furnishings and equipment. Again, think about COVID and the clean down and hygiene procedures that were necessary there to break the chain. Unbroken skin and the lining of the mouth, throat, gut and airways all serve to provide a barrier to infection. The cells of these linings and the substances they produce are the body's first line of defense. If a microorganism does manage to cross this barrier, the next line of defense is the immune system. Whether or not an infection occurs depends on the outcome of a contest between the microorganism and the immune system. The outward signs and symptoms of disease, such as fevers or rashes, are as a result of this contest. Some people may be more susceptible to infection than others, immunity, the system, and those with reduced immunity because of pre-existing illnesses. Some people may be naturally immune to diseases. For example, they may be because they had the disease as a child or else have been immunized. So there's those aspects as well in terms of whether vaccinations, immunization can be used to try and reduce the impact or indeed stop diseases or conditions from taking place. couple of examples on the right hand side there, just to give you case examples to support this. So top right there, Legionella, the water in the cooling towels treated to kill the Legionella bacteria that can cause the disease. So it could be typically ultraviolet light or temperatures outside of or above 58 degrees. So raise the temperature above the ability for it to live in those environments. And then at the bottom, a cleaner of public toilets wears protective gloves and washes their hands when finished to prevent infectious microorganisms being transmitted from their hands to the mouth. So it's not just about the fact that you're wearing gloves, but the body's typical response of, let's say I've got an itchy nose or I need to scratch my face for whatever reason it may be, it can be on the surface of the gloves. So gloves are great as a barrier, but if the microorganism lands on the gloves itself, it's just like it's on the hands anyway. Again, consider the controls that are expected for COVID and the cleaning regimes there to prevent that transfer. So there's a health and safety guidance notes for managing the risk when handling deceased. Like I was saying, I've done quite a bit of work on this area, particularly when considering the mortuary and embalmers and making sure that controls are put in place for that. But nevertheless, there's a decent pathway, should we say, for infection to take place in the transfer of the handling of deceased. So we put in place a risk assessment to consider the impact of potential infection, and then thought about how much controls. And it's interesting in that environment, should we say, where embalmers are doing their work, they have this reluctance to want to have extraction systems on, as an example, in close proximity to where they're working. They have this reluctance to want to wear face masks because some strange people that do this job, but they do like to get personal, should we say, with some of their clients, the deceased themselves. They talk to them in some strange ways, and it just makes me wonder, you've got to be a certain type of person, and maybe they do this because of their personal resilience and their ability to be able to switch off to other things that are going on and treat it in a clinical fashion. But they do create some great results. But you've got to think all aspects. It's not just behind the scenes. What about front of house? What about transfer to the funeral? What about handling of the deceased at the funeral itself? And then things like, going into the furnace afterwards if it's going to be ashes to ashes. So all aspects of the handling itself of disease needs to be considered ultimately to try and control it or indeed eliminate the potential risk. Further take that to another example, primary care. So how can infection prevention and control be put in place for that? Explore the rationale, reduce the risk, challenges that may be around primary and secondary care, emerging and re-emerging infections, and also planning to meet these challenges. So it's quite a bit of research been done on this. 8.2% of patients develop a healthcare associated infection not present or incubated between, but acquired during healthcare exposure. 30% are preventable. NHS, it costs them billions a year. And interestingly, prior to COVID, there was one of the researches that came about that less than 38% of NHS staff were immunised prior to COVID. And I'm not just saying specifically to COVID, I'm saying the immunisation programme in general. Yeah, so I'm not 100% sure as to the reason behind that, but I'm assuming that the belief is that it's invasive. They don't want these chemicals inside their bodies. They don't believe in vaccinations, and therefore it's avoided unless it's absolutely necessary. And think about, when COVID and the government tried to say people who are in direct contact with handling COVID patients must have, and that's what they started off. You must be vaccinated, but interestingly, they've backed off, realizing that they couldn't change that culture, and they couldn't do it without impacting on the number of available staff to be able to treat. So it's always made me wonder why that is the case. Why is there that attitude, should we say? So approaches, and it's typical of most areas of biological agents. Hygiene on the hands, PPE and protective clothing and extra layers that are disposable. Management of that waste, not just the clothing itself and the gloves, but also the materials, the stuff they put on the beds, the disposable tissue papers, etc., etc. So management of waste is a big control management there. Body fluids, and thinking about when they do come out of the body, how are they contained? Do they stop them from being a spill onto surfaces, pores, etc., and small amounts of body fluids that perhaps soak into other materials. So again, cleaning regimes are very important around the body fluids. Management of sharps. So if you think about it, sharps that are used on the operating table, sharps that are used prior to operations, and then the contamination tubs that they have for sharp use, so that once it's gone into a plastic tub, it's then treated as special waste. Decontamination of the environment and the equipment. So there's quite a few different sterilizing substances that are used, but hot water, boiling water, steams are also quite effective in this area as well. So it's like everything goes into a giant dishwasher, as an example. Typically, what we're saying is infections of temperatures above 58 degrees are significant enough to kill off these viruses and bacteria in the air and on surfaces, as well as detergents and bleaches and the variations that they have on cold sterilizing substances as well. And then personal health issues being checked, medical investigations, etc., to keep, and health surveillance to keep an eye on people who may be vulnerable and susceptible. So let's look at some examples. The typical approach that's put out there as education for the NHS staff and care staff follows this rule of a good hand wash, where your hands allow the opportunity to rub all the surfaces of the hands, and it takes typically 20 to 30 seconds. But rather than just a quick hands coming together, dipping into the water, the running water, etc. So that movement of the rub itself of the hands is very important to consider contact with all surface areas of the hands to remove the potential agent. Alcohol rubs, but again, the rub there, it means less time requirement, so that was preferred in some respects, but there are limitations on alcohol rubs, and also some people react to that as a content. World Health Organization put together some advice on hand hygiene, and that's been applied through COVID. And the thought process around before handling, before intimate, shall we say, close contact tasks, after body fluid exposures in their various types, after contact, and then also after contact with patient surroundings. So making the area sterile as well. So all of those 5 areas have to be considered, and you can see now how some people perhaps didn't do as much as they should have done, because it's quite a habit to develop to such a high standard. There we have it. There's the typical example of the advice that was given to NHS staff in terms of the proper way to do hand washing. But they also did some investigations into areas that are more exposed and potential, but also if you can be missed, should we say, during the detailed hand wash. So most frequently the missed, as you can see there, around the thumb on the back of the hand, and then fingernail areas as well on the back of the hand, and then in between the fingers as well. So if they don't do a decent rub, there's potential infection transfer. I've always said this, and that's why I thought it was worth put in there. When you have a regime in place for clean downs, quite often we'll focus on barrier creams to act as a barrier and emulsifier, so that once you go into the workplace, there's less chance of getting dried, cracked hands. So fair enough. I do have concerns about barrier creams, so that's available on one side of the wash unit. Then in the middle you've got the soaps, so you're going to clean, sterilize and remove bacteria. But then on the final side, I strongly recommend this, is on the right hand side is usually moisturizer. So what you're doing there is replenishing the skin with the oils that have lost during the amount of washing that takes place. So if you're over cleaning, which takes place a lot with people, you can get with skin conditions, dryness, cracks, but also dermatitis, because of the amount of washing, particularly in care homes and in NHS. Yeah, so literally 5, 6, 7 times of cleaning can take place in under 10 minutes. So if you're doing that over the hour, and then if you're then doing that over the day, you could literally be washing your hands 150 to 200 times a day in order to keep the high standards of hygiene. Therefore, please remember the importance of moisturizer, replenishing the oils in the skin afterwards. Otherwise, dry, cracking and conditions concerned. Personal protective clothing, all aspects of this, that extra layer or barrier to prevent contact, and also in some cases like COVID, breathing in, so respiratory protective equipment as well. And there you can see examples: gloves, aprons, goggles, masks, worn when needed, and the higher risk groups would require higher levels of protection. And then below that, you might have uniforms, and there's an area of question. Well, where do they get changed? Where do they wear uniforms? Sometimes when they're wearing uniforms, should they be wearing PPE, disposable PPE over the top of it as an extra layer? And that's something that's questioned quite often. And you could argue that it's lack of availability in some respects, but also habits that have formed rather than putting that extra apron on or extra disposable apron as a layer, was opportunity missed. But also think about things you put on the patient as well. It could be disposable large pieces of tissue, or it could be like a disposable apron, whatever it may be in its small set. Lots of different things are out there on the market now to try and control hygiene around interaction with patients. So, as we touched on earlier, hazardous waste is a controlled, so clinical waste comes under this, and it has a special system so that you can keep it segregated and controlled. So typically, from my experience working with the NHS side in the university and in the medical side, what we'd have is our nurses that were going through training, and also the facilities department that were assisting in the management of waste. So I had to go through a training program to explain the handling of the waste, the control measures, the double bagging that might be necessary, the lockable bins that need to be in place, and then those lockable bins being put inside another special area that might be behind lock or key again, so that it reduces the potential for people to interfere with it. And think about what I was saying before about special wastes like sharps and the handling of that, etc. Yellow bags and tagged off at the top, labelled adequately, etc. So there's a lot involved in that, and the information and guidance is available linked to the hazardous waste regulations and handling of clinical waste. Sharps, typically, if they have vaccination programs, they might have hepatitis vaccinations to try and control that, but you can see there the risks that are associated with or higher risk exposure. So hepatitis B is the chance of 1 in 3 if you get a sharp injury. There's a chance of picking up hepatitis C is 1 in 30 from a sharps injury, and then finally 1 in 300 for HIV. It's affinity, should we say, to propensation of the body contamination. So sharps rely on injection into the skin, cutting through the outer skin, allowing the transfer to go through that upper layer, breaking the skin, and then leaks into the system and transfer to a systemic effect. So sharps are important to be handled effectively and controlled. These, I call them litter pickers. So if you can imagine, you've got litter picker grabbers, so that people who have to remove waste or handle the waste shouldn't be coming into contact or breaking the skin. There's the handling of bags. Even though the thick bags, if you've got a sharp sticking out, the needle could cut through the bag, and then if you're carrying it down by your side, it could break through the skin. So some would argue, can you put in place injection resistance type 5 category gloves and the likes of? Well, yes, but does it cover the body? And what about the manual handling activity, etc.? So all of that needs to be looked at in terms of the potential risks. So as you'll see with the sharps box there, it's how it's transferred from the hand into the box and who takes control of that, responsible person, and then thinking about the procedure of where it goes, how often it's removed. Clinical environments do this without question, but it has been interesting in terms of educating some people in a university environment, how they will perhaps, unless they realise this habit needs to be formed from early days, how it can be missed and in some respects bad habits developed. So some general points there. It's the user's responsibility to discard it. So if you can imagine, I'm administering an injection to someone in, let's say, in a ward, then as soon as I've used it, it needs to go straight away into the container. Dispose it immediately. Dispose as single unit. You know, it doesn't mean take bits apart, because if you are fiddling about with it, there's a chance then that you can puncture the skin. So put it all in there. Don't pass it from hand to hand. Keep it in one hand. Keep it contained and controlled effectively, and get rid of it as quickly as possible. Use the approved bins. Correctly assemble the bins. Quite often people leave the lids off, which is, again, contravenes the standard. Temporary closures are in use as well, and they can get broken quite easily. Consider the regulations around handling of sharps in the EU. We don't have to comply with it, but it's just good safe practice. And then it's all controlled by a risk assessment, but more importantly, checks that are taking place, typically through audits and inspections, to show evidence that you are compliant with not just the regulations, but the practices and the policies. If someone is in exposure, should we say, to this, the approach typically that we have is wash it, bleed it, and report it to a nurse manager and GP. So that's the advice that's been given. Wash it, bleed it. When we say bleed it, try and force some of the blood out of that might be infected by contact with the sharp. So just pinch it to force some of the blood out, and that might be enough to take it away from the body rather than absorbed into the body. So that's the general idea there, but a reporting procedure that supports it to make sure that when you do get a needle stick injury, that it is on file, and then if someone does suffer an infection later on, it can be traced back to that particular incident. Decontamination in the environment, in the use itself, and the potential for infection and cross-infection. Obviously, high standards of cleaning, disinfection, and sterilisation are required there. So think of an operating theatre. You would love it to be perfectly sterile. Unfortunately, it's not that good, because in some hospitals they haven't invested the time and effort to get it to such a high standard. So there is still that potential for people to pick up infection during operations. Again, sticking to this idea of other bodily fluids and excretions that come from the body, the fecal specimen should be obtained as soon as possible following onset of symptoms of diarrhea. From my experience, it's about the gyms and the swimming pools that I work with, and some of the overchlorination that we might have to do, or the removal of the fecal inside the water and containing that area, treating the pool, or literally if it's in solid state, you just need to just apply a little bit of cleaning. If it remains solid, you don't have to do as much. So the variety, if somebody has diarrhea, then obviously the pool has to be evacuated and overchlorinated for a period of time of, say, half an hour before people can go back in. And we also do a back clean as well on the system to make sure everything goes through the filters. So you can see the propensity for infection if it is things like diarrhea and the controls expected. So someone who's got an upset stomach shouldn't be going into a swimming pool. Things like that need to be considered, particularly in the care environment, because diarrhea and fecal specimens happen a lot in vulnerable people. So control measures are expected. Gather all your equipment, think about how it's going to be cleaned down, think how you can maintain a clean and sterile area, think about the patients themselves and the respect that they deserve, possibility of leaks that might take place and absorbent materials that need to be contained, and then in some respects, what is the diarrhea? What's the cause of it? Does it need to go to a lab? How are you going to get that checked out? So salmonella is a classic example there. Food poisoning that's resulted in diarrhea, and therefore it may need to go off to the lab to be tested to confirm that it is. If you've got an outbreak, should we say, in a care home, it could well be related to, I don't know, the meat in the pie, as an example, that's been allowed to build up the bacteria levels inside it, and then when it was eaten with several people, they're all showing the same symptoms, and therefore it needs to be tested out. So there's procedures then for obtaining these aspects linked to diarrhea and other forms of discarded, should we say, body waste. So how can we contain it? How can we handle it? What do we do with it when it's being transferred? Thinking about stopping further spills and contamination controls there, all the way through to policy, procedures, training, instruction, etc., and then clean up afterwards. So it all's got to be managed just as you would with a risk assessment process. Mnemonic protocols could be applied when managing suspected potential infectious diarrhea. So SIGHT, S-I-G-H-T, is used. Suspect, isolate, gloves, hand wash, and then test. Yeah. So classic standard that's used inside care homes for controlling and thinking about the potential problems of handling an interaction with diarrhea, but it's a protocol. Adequate resources so you can do the job. That's basically what it's saying here. So thinking about how spills can form, body fluids, what needs to be done to clean it up, what do I need to do to and have access to clean up what's needed, and then making sure that it's left in a sterile state afterwards. So all aspects around the equipment and in the environment that's being used as to how you control and reduce the risks. There's a green book, as is known in NHS, for vaccinations produced by the Department of Health. And the idea comes up infection control in regard to the Health and Safety at Work Act in the generality, but it then goes into a lot more detail. And if you look at that example in the contents page on the right-hand side, you can perhaps appreciate the detail that's involved in that procedure. 455 pages of a document there. So in care homes and in NHS, they have to drill down all of these areas, and they have to consider the policies and procedures that are going to control and provide safety in the aspects. However, part B focuses on the different types of diseases and conditions. So a big list there, all the way from anthrax down to yellow fever. And that's why it's so detailed. Now, I'm not saying you need it, but what I would say, not for the purposes of the diploma, but what I would say, it's very useful to have access to that to appreciate the different types of diseases and perhaps the vaccine regimes that might be considered to reduce or control that risk. They're not without risk when you do vaccines, and because it's putting chemicals into your body and therefore it can affect your immune system. But nevertheless, there's a heck of a lot of detail in there. It's a useful document if you're regularly involved in managing the risks of biological agents. Direct patient care, so the staff involved, routine immunisations that should be in place, he says, tongue-in-cheek. MMR especially important to prevent transmissions to vulnerables, hepatitis B, in contact with blood or blood-stained body fluids. I had to do that when I was in the prison service, again, because hepatitis B was prevalent, should we say, in some of the inmates that came into the prisons. And then you've got varicella for susceptible HWCs who have direct contact with patients who are infected. As I said earlier, we'd like to think that most people have vaccinations in place and are doing what they should do, but surprisingly, the statistics show that it's nowhere near where it should be. So you could argue this was done just before coronavirus came in, but at that time there was a few emerging challenges. So that's why I've put down at the bottom and now COVID-19. And again, from some of the examples that were listed in the video you saw earlier. So a lot of people say, is there an issue around, evolving viruses and the super viruses and the likes of? Perhaps there are. It comes down to the body's immune system, susceptibility, conditions, more contamination in the environment. So much can affect this, but nevertheless, it's an interesting area to study. So what they do as a government is they look at specific areas around antimicrobial stewardships, and they have this mnemonic target: treat, antibiotics, responsibly, guidance, education, and tools. Yeah, so adequately resourced, training people up, giving them that responsibility to make it happen, and then also discipline procedures when it doesn't go down that route, but resourcing them adequately as well so they can treat the infections, but also antibiotics are in place where necessary. And that not just limited to one area, it also tries to target improvements in society as well. Understand the challenges. MRSA was as an example of this. Different mechanisms of transmissions. Quite often it can be complicated, etiology, and therefore propensity for infections to then spread becomes problematic. So we have to think about, the areas, the concerns, the chance for it to get into the body, and that's trying to break the chain of infection. In the NHS, they've come up with medical standards, and you can see some examples there, and they are audited against these medical standards. And their reports are produced, and they have targets they're trying to achieve every year, and as a result of that, it can have impact on budget and resources and identify areas, hospitals and primary care outlets where they aren't quite as good as they should be, and therefore they can be investigated further and action taken. What they've identified from the audits are areas where they could make improvements. So not as good as they should be in certain areas. Cleaning could be better. Infection control could be better. Management and handling of sharps is a major concern. Not in compliance with the sharp handling regulations, as I said before. Policies and procedures not good enough, and then the training that follows, and then the evidence that they've looked for in the audit and the inspections, again showing deficiencies in a variety of different areas. stop that there before I finish off talking about coronavirus.